real-time application of bioinformatics. What is bioinformatics? What is the scope of this particular subject and where it is heading in the current scenario? I think Professor NBR has made my job little easy, because he might have specifically delivered a talk on the genomic medicine and other associated aspects. So let me go into the fundamental details and the real-time applications of bioinformatics in various fields. Specifically, let me start from the basic. What is this bioinformatics? So as the word itself, the bioinformatics itself is going to give you a very specific idea. You know that two words, it is a combination of two words. One is biology and another one is informatics. And it is an interdisciplinary subject. So informatics itself is an interdisciplinary field where you are going to see the computer hardwares will be there, and then computer software and networking, internet. So many aspects of computer science and technology is going to come under informatics. But we are utilizing the informatics technology. We are adopting the informatics technology in the field of biology. Specifically under the purview of bioinformatics, it is purely the molecular data that we are going to handle over here. And also, as I said, it is an interdisciplinary field. The molecular biology, that is, we are talking about the data in terms of DNA sequence, RNA sequence, amino acid sequence, protein structure. And so it is, especially the post-human genomic period, we are seeing in the past 1 or 2 decades, there is a very huge leap or a development in technology, especially from we are there currently now in the third generation sequencing. In other words, it is called as the high throughput sequencing or next generation sequencing, whatever the name is given. A lot of advancement in terms of the facilities and the instrumentation that is available for sequencing. Very rapidly it is developing. So specifically, it is the biological data that is being generated in huge GB, and then it is, that is why it is called as the big data. So kindly mute from your side, otherwise it is going to disturb. Crosstalk. So it involves developing algorithms, databases, and software tools to understand biological processes at the molecular level through the computational methods. So what is this data? I'm talking about storing of the data, accessing of the data, analysis of the data, upgradation of the data. So mainly the biological data, when we are going to address the field of bioinformatics, informatics itself, it is an information repository. So what are the different types of data, scope for handling different types of data under the purview of bioinformatics is, it is the DNA sequences, the nearly more than several decades we have been working and focusing on the DNA, and the very development of the Sanger sequencing technology, where nearly 300 to 400 base pairs, people were struggling in the lab for days together in order to sequence around 400 to 500 base pairs. That itself would have been a very great achievement. But in today's scenario, within 24 hours, we are able to sequence the entire organism's complete genome. We are able to sequence. And if you are going to visit the Human Genome website and their archives, and then if you are going to see what was a $3 billion today, it is almost in Bangalore, if we are able to send the blood sample today, within 48 hours, we are able to get the link to the entire genome of that particular sample. And another 24 hours, if you are going to give completely annotated human genome data, we are able to get somewhere for around 40,000 rupees today. We are able to sequence the entire genomic sequence. So the DNA sequence is the fundamental data where we are handling for the analysis. And over that, the protein sequences, the amino acid chain that forms the protein responsible for the various cellular function, a brief description I have given over there. And of course, today we are there in the next generation sequencing. So we are talking at the level of the whole genome sequencing, whole exome sequencing, and also the specifically the targeted sequencing. So these are the different levels where we are going to sequence the complete genomic data of an organism. And then the genomic data can be converted into a functional genomic data or a structural genomic data we will be able to convert. Or on the other hand, in today's scenario, from a single tissue at a given point of time, all the protein that it is going to express at that particular point of time, that is also specifically considered as a proteome data. Or on the other hand, another way is identify all the genes that are expressing in a genome and then convert that into the protein that it is going to express. That is also specifically called as a proteome data. So these are the different types of data that can be handled. And of course, now ultimately, wherever, whether you start with the DNA or whether you start with the amino acid sequence, you have to talk about the function of that particular gene. So for that reason, we should know the pathway to which the gene is associated, the product of the gene is associated. So thereby the metabolomic data is of utmost important, and accordingly we are going to see the databases that are specifically developed. In order to identify the gene and its function, we are going to do a number of software tools we are going to utilize, and we are going to do the sequence analysis. So under the sequence analysis, the first type of analysis that we are going to do with the sequence is the pairwise alignment, multiple sequence alignment. Under pairwise alignment, we are going to do the global alignment, local alignment. So these are the different types of analysis in order to identify the gene that we are investigating or the protein that we are investigating. So again, if we have to see what is the evolutionary relationship of a set of data, what is the line direction of evolution, and how the say the organisms showing identity or a similarity with the protein or the gene, how are they evolutionary related. So for that we are going to do the phylogenetic analysis. Then the most important analysis that we do is in a given fragment of DNA, whether the coding region is present or not, whether the gene is present or not, or exon is present or not, and what is the exon, to what protein that it is going to express. So in that direction, gene prediction is a very important step. So we will be able to use the NCBI ORF finder or the other Augustus and various other softwares are there. So these are the different types of tools that we will be able to use. For eukaryotes, we will be able to use a different tool. For prokaryotes, we will be able to use a different tool, and thereby very accurate prediction of the gene, the location of the gene in the given sample has to be done. So this we will be able to take the support of the databases also. So the NCPI database or the DDBJ database or the EBI database, we will be able to take the support, or UniProt database, we can take the support and identify the gene and what is the product of that particular gene by using the BLAST10, BLAST-P or the FASTA. So these are the database searching tools we can use, homology database search tools we can use, and then we will be able to predict the gene and also the identify the gene that the DNA fragment is expressing. Then another important step that we will be able to do the analysis is the gene expression analysis. We have to very critically see that what is the gene, what is the protein that a particular gene is expressing, and then what is specifically the pathway that it is associated, what are the protein-protein interactions that it is going to undergo, or what is the post-translational modification that a particular protein is going to undergo. And finally, we have to identify the domain of that particular protein. If we have to identify the protein that it is expressing, and also the functional motifs a particular protein is having, then prediction of the secondary structure of the protein has to be done. Then finally, the 3D structure prediction by using the PDB database we will be able to use, or where the experimentally X-ray crystallography or NMR methodology is used for the prediction of the 3D structure. And if a experimentally determined structure is not there, then we will be able to go for the homology 3D structure modeling using the Swiss model. And now, of course, the machine learning and artificial intelligence is the buzzword that we are hearing in almost all the area. So it is also integrated in the structure prediction tools. AlphaFold is the latest software where we are using for the 3D structure prediction of the protein. So this is a very brief overview of the computational techniques that we are going to use in analyzing a DNA sequence, or the RNA sequence, or the amino acid sequence. Then, of course, the digital libraries, known annotated data storage is of a great help. So the biological databases, by the type of data, if we are going to classify the biological databases, basically based on the sequence databases, we will be able to classify which are going to store specifically the DNA sequence and the protein sequence. So I'm going to online itself, I'm going to specifically show you the different databases that we are routinely we will use at the fundamental level. You will be able to see and this is the homepage of NCBI database that I am showing over here. And if you are going to pull down all databases, beside this there is a pull down menu. So if you are going to click that, you can see how many databases are there under the NCBI. NCBI is mainly a very huge research organization, and they are maintaining under the National Library of Medicine. In association with that, they are maintaining nearly more than 3 dozen databases they are maintaining. So for example, if we want to query some data, so let me select a simple protein over here. So if I want to identify the nucleotide sequence of the insulin, so here you will be able to see that within seconds, sitting here in a remote place in India, I am able to log in to a server that is located somewhere in US, and then within seconds, within a fraction of a second, not even a second, I'll be able to access information from that particular server. You can see over here the count that nearly around 24,000, say 2,42,227 records are there on insulin. So specifically, if we want to see the insulin gene from Homo sapien, here you will be able to apply a filter, and then you will be able to look at the data over here. So here we have a Homo sapiens insulin, but it is a partial CDS sequence that we will be able to see here. So if you are going to click on the data over here, you will be able to see the data in the form of GenBank format. So GenBank is the universal heterogeneous database where data from all the organisms, whether it is a eukaryote or a prokaryote, the DNA sequence is allowed to submit to the GenBank database. It is a universal repository of DNA sequence, nucleotide sequence, and systematically you will be able to see the fields over here. So it is the locus line is going to carry the accession number and the size of the data, how many nucleotides are there in the data, and what is the topology. Since it is a CNA data, the molecule will be given as mRNA, and the topology is linear, and on what date the data is submitted, you will be able to see over here. 25th July 2016. So the title of the experiment and the papers that are published, and the annotation is going to start in the feature table. Features you are going to see over here. In the feature line, we have to fill the, when we are submitting our new research data, we have to fill all these fields in the submission data submission software called as Webin or Sakura. We will be able to use and submit the data to the database, our new research data. You can see over here 1 to 20, 285 is the number of nucleotides over here, and the gene is the whole length. The 285 nucleotides are forming the entire length of the gene, and the coding region CDS is also from 1 to 285, and the translation of that sequence into the amino acid sequence you will be able to see here, and the functional side, signal peptide side, and other miscellaneous features we will be able to identify. In the gene symbol is INS, and at the bottom of the record you will be able to see the actual DNA sequence over here. So this is how systematically all the records, all the sequences that has to be submitted to the GenBank database, systematically in the same order we have to submit. Then apart from that, we are having other formats of the sequence. So whatever I'm showing over here is the GenBank format. And for the analysis, we will be able to say omit all the other details of the sequence, and we will be able to take the data only in the FASTA format. So you will be able to see here what is the FASTA format. FASTA is a type of computer format where almost all the softwares are going to accept data in this particular format. You're going to see in the first column, first line, there is a greater than symbol, followed by the accession number, and then followed by the organism name, name of the gene, and then the molecule, whether it is a DNA or a mRNA, and then whether it is a complete gene or a partial CDS data, you will be able to see over here, and all other information fields are masked over here. Only the sequence you will be able to see here. Then apart from the FASTA format, you are also able to see the data in one more format, that is the graphic format. So graphic format is going to give us an idea of where exactly the location of the gene is there in a graphical way, and specifically you will be able to see the location of the gene over here, and complete information you can see over here, IGF, insulin, and then you can see the location from where to where it is located, length, and all the other details, say the blast protein, blast to genome also we will be able to do here, and then the GenBank record accession number you are going to get. So this is how systematically we are going to see the significance of the databases. Why do we need databases? Databases or repositories, digital repositories where completely analyzed and annotated data is available. So this is how in a data in a sequence database is stored. And then coming to the structural database, the especially the protein 3D structure, it has to be stored in a separate database, because the very definition of a database, it is a collection of a homogeneous or uniform way. We have to, it should have a homogeneous set of data. We cannot dump everything in the same database. So specifically, DNA sequence should have a specific DNA sequence database, and again similarly the amino acid sequence, it goes for a different database. We cannot mix the data over here. Similarly, the functional databases and also the structural databases. So in detail at the end of this particular session, I will be discussing about almost all the data, different databases depending upon their classification. So structural database, the best example for structural database that we will be able to give is the PDB database, MMDB database. So these are the different types of data. So if I had to show you the structural database over here, So you will be able to specifically come to the PDB database. So you can use the Google, and if you are going to type the PDB database, it is you are going to get a link over here called as the RCSB PDB homepage. So Protein Data Bank, the expansion of PDB stands for the Protein Data Bank. So this is the one and only repository for the protein 3D structure. So the same insulin only I'm going to type here. So we are going to get the 3D structure over here. You can see here nearly there are around 11,408 structures are there. So if you want from different organisms, so specifically if you want only from the human, you can use the filters that are given over here. So many filters are there in order to reduce our load, and what data we want to that accurately we will be able to scale down. So specifically, you will be able to see the structure of the insulin over here, and one line description you are going to get. If you are going to click on the accession number over here, you will be able to go to the complete record of the PDB data here and complete information regarding what is the experimental methodology that is used here. And also you will be able to online itself, you don't have to have any plugin software over here. You will be able to specifically see the structure in online only. You will be able to view the structure in 3D, that is the 3 dimension. So you can see the three-dimensional structure of the protein over here. You can rotate it, and then you will be able to view whether what type of folding the protein has undergone, whether it is a alpha helix or beta sheet, and what is the domain and the motif that it is going to have. And parallelly, you will be able to see the sequence of this particular protein is also visible, and other details we will be able to see here, and accordingly we will be able to select and then view the protein 3D structure online without any plugins software. You can see the beta sheet over here, and whatever spring-like structure that you are going to see here, it is the alpha helix and alpha helix to loop. You can see over here beta sheet to alpha helix. You can see the connectivity, either a loop or a coil. So this is how the 3D structure of the protein we will be able to very well visualize online itself. So the structure databases are going to play a very crucial role. So here you will be able to see here what is the role of bioinformatics in genetic research. So the transition of cell biology to molecular biology in the timeline, if we are going to see as technology progressed, what we were viewing at the cellular level, then later on, because of the development of the microscope and other devices, we could very clearly see the ultrastructure of the cell and also the chromosome. We will be able to specifically in karyotyping, we were able to count the number of chromosomes that are present in the nucleus and arrange them according to the size, and then count them. So similarly, later on, from the, say, specifically whatever we were studying at the chromosome level, later on, because of the availability of the Sanger sequencing, developing and standardization of the Sanger sequencing, sequencing of the chromosome became very possible, and we could study fragment by fragment of the nucleotide sequence data. We were able to generate, and we could identify whether a gene is located in that particular fragment of the chromosome or not. And this is how slowly the direction of research started moving ahead. And today we are there at the level of genomic sequencing. And by comparing a healthy person sample with that of a say disease clinically diagnosed disease express person sample, when if you are going to compare, we will be able to very clearly identify whether if there is a mutation in the patient sample, which gene is mutated in that particular patient, and how the protein is getting modified, and how the pathway is going to be altered. So we are heading in a very direction wherein we will be able to use the genomic data, whole genomic data or the whole exome data. We will be able to use it for diagnosis of the disease accurately, and at the same time we can identify the variation mutation that has happened, and thereby correlate that mutation to the disease by analyzing the protein over there, and thereby one will be able to design or think about a very highly specific line of treatment for that particular patient. So this is a overview that I have given the role of bioinformatics in genetic research. It is really contributing in understanding the normal pathway at the molecular level, and identification of the genes, and also understanding their function. We will be able to, by knowing the gene and its expression and the pathways that the gene is associated, the product of the gene is associated, we will be able to understand for a healthy living in a healthy person, what is the normal pathway, what exactly are the protein interactions that are happening, and thereby, if at all, if any of the genes are associated in that pathway, if they are mutated, what will be the impact of mutation, and is it going to result a disease, and in what way the pathway is going to get altered. So this way, the analysis of the say the sequence data using the databases and also the various softwares that are available, we will be able to identify and what is the end result. Here I have taken the human genome as an example, and thereby in that direction I am discussing. So the same protocol can be applied for the genome editing of any organism or plant, in order to increase, in order to make it pest resistant or disease resistant, and thereby one will be able to increase the yield or horizontally transfer any useful gene into that particular genome, and one will be able to say work at a customizing the genome. So here I have taken the example of the human genome, so that is why I have given here the especially the softwares and the database in whole, the concept of using application of bioinformatics in genomic research. It has a very immense implication in terms of personalized medicine or preventive medicine, with the great information, with the genetic information. We can guide to tailor-made treatment plans. Especially we are seeing, if you are able to recollect, say 3 months back, especially the IIT Bombay and specifically the Tata Memorial Cancer Hospital, they have joined hands for gene therapy, so that in a cancer patient, very critically his genome will be analyzed, and for the type of cancer a patient is suffering, whether a marker, genetic marker gene, if it is responsible, then the mutated gene, can it be say specifically replaced with a healthy gene copy by the gene therapy. So this is how the whole direction of research is getting modified. So as I said, bioinformatics is an interdisciplinary field. You will be able to see what are the fields that are going to come under the umbrella of bioinformatics. Is you can see here, of course, biology is the fundamental, especially the molecular biology is the fundamental subject. To this, one will be able to link the microbiology, biophysics, and then physics, and then biomedical engineering, and then biochemistry, genetics and genomics, you will be able to see neurology, biostatistics, computational biomedicine, chemistry, and then mathematics and statistics in order to write the develop the specific algorithms in the software. And of course, you can see the system biology and also the computer science. So coming to the types of data, already I have discussed what are the different types of data that we handle at the under the umbrella of bioinformatics. So we will be handling in today's scenario diverse range of data types, each with its unique characteristics and application. So here you can see the DNA sequence, RNA sequence, and the protein sequence. And of course, you know that the nucleotides are of four types in the DNA. The order of the nucleotides, the combination of nucleotides, they are going to form the whatever the genome, whether it is a prokaryote or a eukaryote. Ultimately, it is these four nucleotides in different combination they are going to form. And of course, the chromosome number is also equally important. And then coming to the RNA sequence, RNA sequences are also specifically composed of, only thing is you are going to have the uracil as a alternate nucleotide over here. Then protein sequences, this is the as per the genetic code, whatever the DNA sequences are there. When you are going to translate a DNA sequence, you will be able to, by applying the universal genetic code, we will be able to convert that into the amino acid sequence. Then coming to the structural data, the classification of the data at the molecular level, one is the sequence data. The second type of data that we are going to handle will be the structural data. So protein 3D structures of protein, including their secondary structure details, or otherwise I'll be able to sum up as the secondary structure and the tertiary structure, and of course the quaternary structure is also taken into account. And nucleic acid, three-dimensional structure of the DNA and RNA molecules, including the double helix and the other complex structures, the data is also handled. And of course, at a larger level, as I said, today we are there at the next generation sequencing, or the high throughput sequencing, whatever we are going to call. Today we are level at the instrumentation is at the level of sequencing the genome. With ease, we will be able to, with 99.9% accuracy, we are able to sequence the complete genome of an organism. And then we will be able to compare. For example, if I'm going to take the human genome as an example, today we will be able to compare. If I'm going to send my blood sample to Bangalore and I want to know what are the variations that are there in my genome, we will be able to specifically compare the candidate's genome with that of a reference genome. So from time to time, the International Human Genome Organization, they are specifically releasing the reference genome. Now we are using the 38 build we are utilizing over here. So thereby, when we compare the patient sample with that of the reference human genome, we will be able to identify what are the variations that are there. It can be SNPs, or it can be insertions or deletions, or it can be copy number variation, or at the chromosomal level, you will be able to see that whether a duplication has happened, or one part of the chromosome is specifically copy pasting has happened, or cut pasting has happened. So thereby at the structural level also we will be able to transposons or reverse transition has it happened. So we will be able to identify all the types of mutation that has occurred. In other words, in one line, if I have to say what are the different types of genomic variations that has happened, and whether that is responsible clinically for the expression of a disease, we will be able to identify. So coming to the transcriptomic data, today we are going to see, if we are going to see the publications over the genomic data. Today we are preferring the transcriptome data because genome is a standard entity that is present in all the cells of a particular organism. But on the other hand, all the cells of an organism need not express all the genes that are present in the genome. So the gene expression is going to change from tissue to tissue, and also age, time, environmental factors, or the nutritional factors that an individual is having. That is why it is called as nutrigenomics. And apart from that, now one more factor that is being added over there is the stress, right? So, in other words, in one word, if we had to say, it is completely the lifestyle of an individual that is going to, by and large, it is going to have an impact on the gene expression. So by sequencing the genome of a organism, we will not be able to definitely say that whether a gene is upregulated or whether a gene is downregulated. So in order to understand that and scale down on that information, it is better that we are going to specifically work at the mRNA, that is the RNA expression level, and then from the mRNA we will be able to convert that into cDNA, and then we will be able to see what is the level of expression of that particular gene. So then coming to the why mRNA data is being preferred over the genomic DNA data is the right type of exons has to be joined in order to form a functional gene. So if we are going to make a mistake of selecting the right type of exon, then what is going to happen? The protein that is going to get translated in the computer, it will be totally wrong. So that is why mRNA, a functional mRNA, in other words, we would have undergone alternate splicing, and it will be ready for the translation process. So thereby, the mRNA data will be more accurate when it is compared with that of the genomic DNA data. So another type of data that you will be, instead of just working on one protein or one gene, one will be able to see all the proteins that are expressed in that particular tissue at a given point of time in a particular environmental condition. So thereby, the proteomic data is going to give us a very clear dimension of what are the proteins that are expressed at a given point of time, and what are the interactions between the protein one will be able to see. Then coming to the protein modification data, information about modifications of proteins such as phosphorylation or acetylation. So in other words, in one word, we have to say we will be able to study the post-translational modification. So coming to the metabolomic data, metabolic pathways, so KEGG pathway database, wiki pathways. So there are so many, say, public domain pathway databases are also available. And at the same time, we are also having certain paid pathway databases are also available. And also we have commercial pathway construction softwares are available. So thereby we will be able to very specifically construct the pathway depending upon the whatever referencing we have done, and over the metabolic and protein-protein interaction information that we have collected from literature or from the yeast to hybrid experiments, we will be able to prove that a particular protein is interacting with another protein, and thereby the metabolic pathways can be very accurately constructed. And also there are very high-end databases are available on the metabolomic pathway data. So metabolomic concentrations is the level of metabolites in a samples, similar to the gene expression. So what is the, specifically if you go to the UniProt database, you will understand which is the tissue that is expressing the protein at a higher level, and what are the other tissue, what is their expression level. So all this map and information you are able to get from the UniProt database. Then coming to the phenotypic data. So we have the clinical database is there where information about patients' characteristics such as age, sex, and medical history, and observable traits in an organism such as height, weight, or disease susceptibility. So if at all, if there is a ClinVar database, if you are able to log in and see, the best example we will be able to give is the DB, SNP, and ClinVar database, and various other databases are available. So wherein we will be able to understand if at all if there is specifically a SNP is there and how it is specifically correlated to the disease expression. So phenotype ClinVar database and DB SNP and OMIM database, they are great databases wherein we will be able to relate the genotype to the phenotype over there and what type of mutation has happened, how the mutation is responsible. We can use it, those SNPs or the haplotype blocks, we will be able to use it as markers for the recognition of the disease. So coming to the different types of data formats that we must, in that format only we have to store the data, or when we are trying to analyze our data, we have to feed the data to the software in specific format. The most common format, nearly 3 to 4 decades, that is 30 to 40 years, we are using the very first format, that is the FASTA format. FASTA format is basically developed by the EBI group. So they have a database searching software called as the FASTA. Different flavors of FASTA is also available there. So thereby, in order to type the data in that software, we have to use a particular format called as the FASTA format. And FASTA format, almost all the tools of bioinformatics, they are going to accept it. If it is a genomic data, then we are going to use one more version of the file format, that is the FASTQ format. Because you know the next generation sequencing is a parallel sequencing. At a given point of time, several strands of nucleotide will be parallelly undergoing sequencing, and the quality test will be done. Q stands for quality test. So the quality test for the sequence that is generated by these next generation sequencing machines will be done, and that will have at the bottom not just the sequence. Below every nucleotide, there will be a quality score will be there. So that is why this format, in other words, you are going to call it as the FASTQ format. So FASTQ format is a next generation sequencing, Illumina sequencing, or any other method if you are going to use. The latest machines, they are going to give the output in the form of FASTQ format. Then from FASTQ is a heavy file, so thereby we have to convert it into different file formats like the SAM format or the BAM format or the BED format. So these are the different formats we are going to use. And one more format that we are going to use is we are going to compare a individual's genome data with the reference genome of human, and thereby all the variations, all the mutations that are there in the individual can be identified, and that is written as a separate file. That is what you are going to call it as the variant call format. In other words, it is called as the VCF. Then for the protein 3D structure, we are having a very specific format called as the PDB format, where the every amino acid that is there in the protein, it will be specifically say considered as whatever atoms that are going to go for the formation of the amino acid, and thereby the every atom of that particular amino acid is going to have a coordinate that is generated by the X-ray crystallography or by the NMR, X coordinate, Y coordinate, and Z coordinate. So thereby these atomic coordinates that are recorded by the X-ray crystallography is specifically written in a particular format if you have to submit to the PDB database. That format, in other words, you are going to call it as the PDB format. And PDB file format we can download, and also we will be able to visualize it in some of the visualization softwares like Biovia Viewer, which is a commercial software, or Schrodinger, or Rasmol. So these are the various, or Chimera. So these are the different tools that are available for protein 3D structure visualization and analysis. So understanding the data types and their associated file formats is essential for effective bioinformatics analysis. So there are various conversion tools are also available. We don't have to panic that if we have to convert from one format to another format, we will be able to convert our data from a particular format to another required format. So thereby analyzing and interpreting the data, researchers can gain insight into the biological processes. And what is the effect over here is one will be able to, if we are going to take the human health as an example, understanding the disease, understanding the molecular basis of the disease is of utmost importance. When once we know what is the gene that has undergone a mutation, or a protein that has undergone a misfold, and thereby that is causing a disease, means that protein can be used as a target for the development of new drug. And thereby, since it is a structure based drug designing that we are going to do, the side effects and the toxicity of the drug can be reduced, and the specific targets that are responsible for the disease expression alone, the drug is going to go and bind, and thereby a better line of treatment we will be able to think. So here are some of the major milestones. So whatever today, whatever we are talking in terms of whole genome sequencing, whole exome sequencing, and identification of the variations, mutations identification. So what are we are going to talk today? It is not a overnight developed technology. So if you are going to go by the say the timeline, we are going to see that over a period of 10 years, I have taken the main milestones that has happened in the field of bioinformatics. So by 1960s, if you are able to see the papers that are published during that time, the first computational tool for DNA and protein sequence analysis were developed. This is like laying a foundation to this particular field, and very at a very fundamental level. And then in 1970, if you are going to see there, Margaret Dayhoff developed the first algorithm for sequence alignment and fundamental technique in bioinformatics, whether at the time they were calling it as computational biology. And later on, because of the, say, Margaret Dayhoff, she developed the database at her institution level. She was the first one to document the protein 3D structure, and it is called as a protein atlas. Later on, she converted that protein atlas into a digital format using the technology, and later on, it was there only at her research organization level. And later, they converted the database using the internet. So anybody from any part of the world were able to say specifically browse the information that is stored in the database. So in 1982 is the major milestone that you are going to see. The American government, they came out with the funding for NLM and National Institute of Health, that is NIH and NLM. They were given a major responsibility because NLM was already associated, National Library of Medicine. By the time 1982, they were digitalizing all the records. So thereby they were given the responsibility for the creation of the NCBI database. So thereby GenBank database was formed. It is a public database of DNA sequence, was created, providing a centralized repository for the biological data. Very specifically, that is why the DNA sequence database of NCBI is called as GenBank. So if we go down the timeline from 1990 to 2003 is the period where the scientists around the world nearly participated in the Human Genome Project. And it is a very, as you know, it is an international effort to sequence the entire human genome. And this project specifically generated vast amount of data and accelerated the development of bioinformatics tools and techniques, especially whatever state of art facilities in terms of molecular biology lab instrumentation, and also the various software tools, and also the software's databases. Rapid transition happened after 2003. Especially the whole genome sequencing machines. Illumina came out with a number of high-end technology machines to the market, and thereby a remarkable development or advancement happened in the area. So early 2000, if we are going to say the advent of next generation sequencing technologies revolutionized genomics, allowing for faster, cheaper, and more accurate sequencing of the DNA. And by 2012, the CRISPR CIS9 gene editing technology was discovered, enabling precise manipulation, and specifically it opened a new perspective or dimension for the possible treatments that are possible using gene therapy in the field of medicine and also in the field of biotechnology. So, if we are going to see the advancements in bioinformatics have led to significant breakthroughs, high throughput sequencing technology and powerful computational tools have enabled researchers to analyze a vast amount of genetic data quickly and accurately. So, this has accelerated a lot of development we are going to see in this direction, accelerated discoveries in the fields such as genomics, proteomics, and molecular biology. So the if we had to really say the advancement in the field of molecular biology and bioinformatics, because all over the world, the scientists they are working on especially cancer genomics. So thereby, in order to find a cure or find a solution for the cancer disease, similarly HIV and other specifically congenital diseases, like the because of the mutation in the gene and that is inherited Mendelian genetic diseases and non-Mendelian genetic diseases, congenital diseases. All over the world in different labs, they are working day and night in order to find a solution or an accurate line of treatment to it. So thereby, whatever technology that we are seeing today, upgradation or advancement of technology that has happened is specifically because of the direction in which the scientists are working. And of course, most of the softwares they are trying to use whatever the artificial intelligence and machine learning algorithms they want to incorporate in the latest softwares of genome analysis. So thereby very accurately the software will be able to identify the mutation that has happened, variation that is responsible for the disease. So thereby the protein structure prediction, drug discovery, and the disease diagnosis. So in all these areas, the machine learning, deep learning, or artificial intelligence is being used. So then now the scientists, they are specifically when it comes to cancer research or any other congenital disorders, single cell genome analysis. That is, you are going to, from the patient sample, you will be able to take a particular tumor cell And in that cell itself, you are going to specifically sequence the entire genome. And from the same patient body, a healthy cell can be taken and its genome can be analyzed and comparative genomics can be made. And then what is the mutation that has occurred for the expression of the cancer disease people are trying to monitor. So it is handling very huge amount of data, big data analysis, and it is a challenge in terms of hardware and software also. So, but now the direction is towards the single cell genomics, where the researchers study individual cells, providing insight into cellular heterogeneity and then the disease expression mechanism. Now we are talking about the metagenomics instead of, for example, in microorganism, from a ecosystem, when you are going to take the sample, multiple strains of microorganisms will be there. When you go for sequencing, if you are unable to segregate the genome strain wise, and it is a mixed data of all the strains that are there in that particular niche, then at that time you cannot call that as a genomic data. You have to call it as a metagenomic data. So the study of genetic material from an entire microbial community had led to the better understanding of the human microbiome and its impact on health and disease. So when the multiple samples are there, and multiple, for example, if 10 people are there in a room and for some reason there is a emergency and you will not be able to segregate the samples over there, we clearly know that the samples are from 10 people, means then you cannot call that as a genome of an individual. You have to call it as a metagenomic study. So similarly, the genome-wide analysis, that is the GWAS studies, is also equally popular. So if we have to identify a marker for a disease, means we have to do the genome-wide analysis study, and then at least 1% of the population who are suffering from the disease should be able to show a common pattern of gene mutation. Then only it can be taken as a marker for a particular disease. So that is also gaining a lot of momentum, Jiva studies. So then coming to the synthetic biology is, say specifically artificially in the lab, specifically we will be able to develop design and construction of new biological system, often with the help of bioinformatic tools, has the potential to revolutionize various industries from medicine to material science. So some of them we use, some of the proteins and enzymes, they are having a lot of industrial application, and also something that is naturally synthesized in a normal cell, because of the amount of that particular protein that is required, or amount of that particular product required in a large scale. So it has to be synthesized outside the cell, biological cell, by using a biotechnology procedure. So thereby, that is why it is called as a synthetic biology. So they are of very high demand in various fields of industry, and also in pharma industry in the form of a drug or in the form of a substituent. For example, if you are going to take human insulin today that is available in the market, it is synthesized outside the normal human cell. So on a large scale, the insulin gene is made to express in a third party organism and the product is harvested. So this is what you are going to call it as. Even the growth hormone and various other proteins are artificially synthesized outside the living cell using the bioreactors or biotechnology. So this comes under the synthetic biology. So, and even now, if you are going to see some of the papers that are recently published, chimeric proteins, you are going to artificially in the lab, you are going to combine multiple domains in the same protein that are having very significant biological function, So one will be able to add those domains of biological interest and then engineer the protein in the lab level. So these are some of the real-time applications that we are going to see. Like this, we will be able to give nearly hundreds of examples we will be able to give. So what I mean to say over here is bioinformatics becomes increasingly integrated with other disciplines. And especially what I want to say over here is molecular biology or bioinformatics is not going to have compartmentalization like botany, zoology, microbiology, or agriculture. We will be able to use the same software in any organism. It can be prokaryote or it can be eukaryote. So we will be able to utilize the same analysis protocol. And the only thing is a dedicated databases has to be there where already analyzed information has to be systematically stored over there so that the support of the already annotated information we will be able to utilize in order to analyze the new data. So that is why bioinformatics becomes increasingly integrated with other disciplines. So even we can expect see even more groundbreaking advancements in the years to come. So coming to, since I'm taking the human genome as an example over here, so bioinformatics is a rapidly evolving field that has significant real world application. So it is not just, we are not talking in theory. Whatever we are going to say that this gene can be replaced by a healthy gene and this mutation is responsible for a disease expression means, yes, inside the cell, in the biological system, that should be. And whatever we are going to analyze and say that this can be cured by using a particular chemical compound means either in our lab studies it may be functional, or in the computer we will be able to say that a particular drug is going and docking with the mutated protein. But in nature, when a tablet is given to the patient, the same drug should be able to, in the actual patient's body also, it should be able to go and bind to the targeted protein only. Then only we are going to call it as a real world application, or in one word we will be able to say bench to bedside. So whatever research is done, it has to be in real time application has to be there, and real world application has to be there. So thereby under that, the very first type of application that we are going to see is the precision medicine. So personalize a treatment plan, we will be able to, by analyzing individuals' genetic makeup, what type of SNPs or SNVs a individual is having. So what type of treatment can be given to him? So what is the metabolic rate? Whether he's an average metabolizer or a fast metabolizer or a slow metabolizer, or that particular compound is not going to be metabolized in his body at all. So that also can be taken, and thereby tailor-made treatment can be given, and dosage fixation can be done as per his metabolic speed, reducing thereby what is going to happen, one will be able to reduce the side effects. So it is something like one rule is not going to apply for every patient. So we have to see the patient's genetic makeup, especially this is very well practiced at the cancer hospitals. So they are going to very specifically see the cause of the mutation by sequencing the cancer patient's genome, and thereby from day one the right type of treatment can be given to the patient. So of course with the development of bioinformatics, and also since we know all the genes that are expressing in the human body and also the proteins, and at the same time from the ClinVar and SNP database or 1000 genome project, so we clearly know that what are the variations and what is the significance of these SNPs or mutations, and thereby the target can be identified accurately. So bioinformatics softwares like the SwissDoc or Swiss model, and also the ligand PubChem, and so many other candidate drug molecule databases are there. So through that, we will be able to identify potential drug targets and design novel drug molecules to predict their interactions with the biological systems. And thereby, what is going to happen? The cost of research is going to come down, and also the time taken to develop a drug is also drastically is going to decrease, accelerating drug discovery and the development process. So not only here I have taken the human health as an example, as per my expertise and domain, but even we will be able to apply in agricultural biotechnology for crop improvement. One will be able to say edit the genome of a plant and thereby bring in the genes of disease resistance, dot resistance, increased yield, enabling the development and improved crop varieties. And ultimately, we are going to see the plight of the farmers. We will be seeing in media, and also we'll be reading in newspapers. So sustainable agriculture is the need of the hour. So by developing sustainable agricultural practices and by analyzing microbial communities in soil and understanding their impact on plant and plant growth and nutrient cycle, one will be able to in 360-degree environmental condition, soil condition. So all these parameters can be taken for better crop yield. Then coming to environmental monitoring, pollution. Pollution is one of the major problem any country is going to face on this particular planet. So analyzing metagenomics is analyzing the genetic material from environmental sample. So one will be able to monitor microbial communities and assess the impact on environmental factors and ecosystem, or engineer the microbial genome in such a way that it will be able to disintegrate or degrade more solid waste or the water pollution. If the microbial tailor-made microbial communities, if they are able to digest the pollutants, then it will be we'll be able to specifically control the pollution. The coming to bioremediation is it can identify microorganisms capable of degrading pollutants. So this is what I said, aiding to the development of bioremediation strategies to clean up contaminated sites. So what is going to happen when we know the microbial genome and thereby how it is going to cause the disease? Then again, we will be able to develop better vaccine and better drugs in order to treat. So thereby coming to the next application will be the forensic science, DNA analysis, DNA evidence. We will be able to treat it as a evidence, the samples that are collected from the crime scene, helping to identify suspects and solve the crime. And then genetic genealogy, by analyzing genetic data, bioinformatics can help to trace ancestry and identify individuals based on their genetic profile. So these are some of the real-time applications that we are going to see, and the list is going to grow for days to come. Almost in every field, the DNA sequencing is being applied, and the analysis of the DNA sequence is of immense importance, and thereby one will be able to utilize it in industrial biotechnology, optimize fermentation process, improve enzyme production, and develop new bio-based products. And then bioinformatics is used in various industries, including pharma, agriculture, environmental science, to analyze large biological data sets and gain insight into decision making. So this is the overall say profile of the studying molecular biology at the DNA sequence level, or at the RNA sequence level, or at the amino acid sequence level, and then seeing the impact of mutation. What will be the impact of mutation on the function of that particular gene, or on the protein structure, and the protein, whether the binding site or the active site of that particular protein, is it getting modified, and thereby in the pathway what will be the role of this normal protein and what will be the role of the protein that is mutated. So thereby one will be able to, not in just one field, in all possible fields, we will be able to apply the molecular analysis. And molecular analysis is possible only when we are having the right type of softwares, and at the same time there should be library. Known information library can be systematically stored and accessed all over the world free of cost. One will be able to access that information. So thereby that is going to act like a say a review of literature, how we are going to do by reading at the papers that are published earlier in the domain where we are doing the research. Similarly, these databases are nothing but digital facilities that are available for us, where the information on the DNA, RNA, and the proteins, and in various perspective, we are having organism-specific databases. For example, Drosophila melanogaster. Drosophila has got a dedicated database. This is called as the organism genomic databases. Potato genome has got a separate database. The silk, Bombax mori, has its own genome database. So organism-specific databases, fly database, or the worm database is there, C. elegans. So this way one will be able to have a dedicated database for a particular organism genome also we will be able to have, and thereby understanding the holistic picture from gene to pathway will be possible by utilizing the bioinformatics infrastructure facility in the form of databases and tools. So I conclude at this level. Thank you. If you are having any question, you can ask.